So here in this video we have this molecular dynamic simulations of an Argonaut 2 protein bound to these RNA molecules. So it binds this orange guide RNA, which guides it to the yellow messenger RNA, of which we only have a small piece. It is usually much larger structure. And with the molecular dynamic simulations, we can observe how the complex is moving around and how the biomolecules are interacting with each other. And even though this simulation is only 100 nanoseconds long, it took a very long time to calculate the simulations on the CSC supercomputer LUMI, because the simulation speed with this polarizable force field with the OpenMM software is very slow. It calculates about 5 nanoseconds per day, so you can imagine how long this simulation took to calculate. And the molecular dynamic simulations are used to study the biomolecular interactions, and with them we can see how the biomolecules interact, and then we can design changes to the biomolecules so they could bind to each other better, or they could bind to each other worse. So if we, for example, would like to inhibit the interaction between this AGO2 protein and the RNA, we could design a drug that would bind there in between the RNA and the protein, and then we would prevent the function of this protein and its RNA binding partner. The pink balls in the picture are the magnesium ions, so this simulation is run with the ions, and it's also run in a water box. But for the visibility, I have also removed the waters here because it's impossible to see the protein from all the water molecules, which are in theory all around the protein. This is to better simulate the conditions inside the cells where the proteins are not in a vacuum.